Today we're going to turn to transcription and RNA processing. We're going to move from the RNA viruses that we talked about last time to mainly viruses with DNA genomes and see how they make messenger RNA. So the red arrow illustrates what I mean by transcription. This is a process specific for DNA viruses. So here on the Baltimore scheme we have our double-stranded DNA containing viruses, the class 1 viruses, and from double-stranded DNA you know we can make messenger RNA directly. So transcription is making mRNA from DNA. You will hear other people call other things transcription, but I do not. I have a specific meaning for it, all right? The making of mRNA from a double-stranded DNA. That's what we're going to talk about today. We're going to talk about the viruses that have double-stranded DNA genomes, single-stranded DNA genomes, viruses with gapped DNA genomes. Those are all DNA-containing viruses that undergo transcription. And we will also talk a little bit about retroviruses. These are viruses with plus-strand RNA genomes, but that genome is converted to DNA, and from it, mRNA is made. So these viruses undergo transcription. They are set apart from all the RNA viruses we talked about a week ago. Those viruses encode RNA-dependent RNA polymerases, which make mRNAs. The retroviruses do not make their own mRNAs. The cell does it for them, as you will see today. Most of the viruses we're going to talk about, with one exception, utilize the polymerases of the host cell in order to make messenger RNAs. It's the rare virus that encodes their own DNA-dependent RNA polymerase. And as you may know, if you've taken biology, there are 3 different kinds of eukaryotic RNA polymerases in the cell, DNA-dependent RNA polymerases. They're shown on this table. They are called RNA pol 1, 2, and 3. And the one that makes mRNAs, of course, in the cell is pol 2. And this is the one that will make mRNA for the vast majority of the viruses that we're going to talk about. So pol 2 synthesizes what we call pre-mRNA, because in many cases it's processed further to make mRNA. In fact, technically, these enzymes do not synthesize mRNA because mRNA has a very specific meaning. It is a molecule made after the processing occurs. So they make pre-mRNA. They also make primary microRNAs and small nuclear RNAs. And for viruses, many viruses, that's the enzyme that makes pre-mRNA for viruses. Also makes microRNA, primary transcripts. And there is one RNA virus that we know of, the hepatitis delta virus genome. This is a viroid, or a virus, let's say, not viroid. This is a virus with an RNA genome that is copied by DNA-dependent RNA polymerase. Now, you may be confused at this point. You may say, I thought RNA couldn't be copied by a cell enzyme. Well, what I said was there's no RNA-dependent RNA polymerase in the cell that can copy an RNA genome. That's why all those RNA viruses have to encode their own RNA polymerases. But this is one exception, and we will talk about this later. Hepatitis delta virus, its genome is RNA, yet it's copied by DNA-dependent RNA polymerase. So somehow the structure of that RNA of hepatitis delta virus fools a DNA-dependent RNA polymerase into copying it. It's the only exception we know of so far. Okay, that's pol 2. Pol 1, of course, makes the precursors to ribosomal RNAs, the RNAs that make up the ribosome. And as far as we know, no viruses need pol 1 to make their own RNAs. Of course, they need ribosomes, right, to translate their messages, but we don't know of any viral pol 1 transcribed RNAs. Of course, that may be proven wrong any day. And then we have pol 3, which makes precursors to tRNAs and ribosomal 5S RNA, as well as small nuclear RNAs, which you'll see today are involved in splicing. And viruses need to make pol 3 transcripts. There are a number of them that we'll encounter if not today, later on in this course. So we're mostly going to be focusing on Pol II. Okay, here's a really important statement for you to remember. Transcription is the first biosynthetic reaction to occur in cells infected with double-stranded DNA viruses. All right? The first thing that happens is they have to make mRNA, because when you think about it, unless they make a protein, they can't start reprogramming the cell to make more virus particles. And as you will see for DNA viruses, a main event is replicating the DNA genome. And you need at least one protein, one viral protein to do that. So mRNA synthesis has to be the first step. Now, this technically is not true for the other viruses with DNA genomes. I have double-stranded DNA viruses on this slide. So if you're trying to guide your thinking, just whenever you see a double-stranded DNA virus, the first thing it's got to do is make mRNA. Now let's say we have a virus with a gapped DNA genome. The genome is not complete. What do you think is the first thing that has to happen? Yes? It's got to get repaired because, again, you have to make mRNA. So no matter what the DNA genome looks like, you can always assume something has to happen to make it suitable for transcription by DNA-dependent RNA polymerase. Now, if you think about that, it's going to really help you guide your thinking about the processes that are happening here. So whether the genome is single-stranded or gapped, it has to be repaired. So technically, the first biosynthetic event in those viruses is repair. But eventually, they all have to make mRNA. So let's take a look again at our Baltimore scheme and think about what we just said. We have mRNA in the middle. The double-stranded DNA viruses can go directly to mRNA. They can be transcribed. Because remember, in the very first hours of this course, I told you the only substrate competent to make DNA substrate competent for the transcription of mRNA is a fully double-stranded DNA template. So the class 1 viruses, adenoviruses and herpes simplex viruses, they have double-stranded DNA genomes. They can pop right into the nucleus and immediately get transcribed into mRNAs. And we'll look at this today. Single-stranded DNA viruses like the parvoviruses, you can't transcribe them. DNA-dependent RNA polymerase will not recognize a single-stranded DNA template. So it has to be repaired first by the cell repair system. It's not going to be repaired, it's not going to be replicated. It has to be repaired, which is a different process, and then it can be transcribed once it's double-stranded DNA. The same for the hepatitis B virus genome, which is gapped, and we'll look at this more in detail later. It has gaps, it has a protein attached to it, has a piece of RNA. There's no way this can be a substrate for transcription. So again, it has to be repaired, and then mRNA synthesis can occur. All right, so that's a key point that you have to remember. Now, there's one other virus we're going to talk about today, the retroviruses. And you may say, well, why didn't we talk about them last time with the single-stranded plus and minus and double-stranded RNA viruses? The reason is because their flow of information looks more like a DNA virus than an RNA virus. Retroviruses have plus-stranded RNA in their capsids. But when retroviruses are up here, class 6, they have plus-stranded RNA in their capsids. When that particle infects a cell, it's reverse transcribed to a double-stranded DNA molecule. And that is now a substrate for DNA-dependent RNA polymerase. Right, so in a way, you can almost think of the retroviral genome as having to be repaired to DNA before it can get transcribed. The RNA is not a substrate for mRNA synthesis ever for the retroviruses. All right, so here's a summary of the repair reactions that we have just talked about. So adenovirus and herpes viruses, their genome is double-stranded DNA. It doesn't have to be repaired. It's transcription competent as soon as it gets into the cell. The adenovirus genome is circular, double-stranded DNA, but it has a gap. It's missing a lot of one strand. You can see that there. It has a protein attached. It's got a piece of RNA attached. Again, cannot be transcribed. It's repaired in the nucleus to form a fully double-stranded DNA molecule, which can then be copied by the host cell DNA-dependent RNA polymerase. Parvoviruses are mainly single-stranded. DNA-dependent RNA polymerase cannot recognize this. It has to be repaired by the host cell. You get double-stranded DNA, and that can be used to make mRNA. And then, of course, the retroviruses, the plus-stranded RNA genome. When this comes in the cell, it is not translated initially. It's hidden in the nucleocapsid of the particle. The ribosomes can't get to it. But in that particle, there is an enzyme, reverse transcriptase, that makes a DNA copy. And that DNA goes into the nucleus. It is integrated into our chromosomal DNA. And there, it is transcribed by Pol II of the host cell. So integration of retroviral DNA is an obligatory step in the expression of mRNAs. It will not be transcribed as a circle. It has to be integrated. And all of these retroviruses integrate their genome into our DNA or the DNA of whatever host cell they're infecting. We will have an entire lecture on retroviruses, but today we're going to talk a little bit about how mRNAs are produced from those genomes. All right, now let's start with looking at the general process by which mRNAs are made in a eukaryotic cell, because this turns out to be in the nucleus where most viruses replicate, with few exceptions. So at the top we have a double-stranded DNA template, a blue and a light blue strand showing the plus and the minus strands. By convention, wherever I show this kind of a red arrow at the top here, that means where transcription initiates, where mRNA synthesis initiates, and the direction of production of mRNAs. So in the nucleus, transcription of a double-stranded template by Pol II results in the production of what we call a pre-mRNA, which is shown there. And these mRNAs are typically capped at their 5' ends. They have an M7G PPPNP. The 5' triphosphate is attached in a reverse direction than normal. We'll look at this in more detail later. The cap is applied to the mRNA after 20 or 30 bases of elongation. It doesn't start with a cap, it's added later. Then you have your pre-mRNA, and as you know, many mRNAs contain what we call introns, or intervening sequences, which have to be removed because they don't code for the final protein. And so that processing happens in the nucleus. It's called splicing, and it's shown here. So your final mRNA has the intron, which is in pink, removed. We'll look at this process in a bit, because viruses utilize this to do a lot of neat things in the infected cell. These pre-mRNAs also need to be polyadenylated. A stretch of about 200 A residues needs to be added to the 3' end. And then that mRNA, after splicing and polyadenylation, is exported into the cytoplasm. You can see it going out of the nuclear pore here. And there in the cytoplasm, the capped and polyadenylated and fully processed mRNA can be translated by the ribosomal translation system to get proteins. So that's a typical series of events for a cellular mRNA, and in fact for many viral mRNAs as well. Now, mRNAs don't last forever. They have a half-life. And this is important because if you think about it, if we made mRNAs and they lasted forever, we'd be making proteins when they shouldn't be made. So mRNAs have different half-lives depending on how badly the cell needs the mRNA. And so they are often degraded anywhere from within a few minutes to hours after they're exported into the cytoplasm. Now, as I said, many viral mRNAs are spliced as well, so they follow the same pathway. But also many mRNAs from viruses do not get fully spliced. So you see here, there is an RNA getting out into the cytoplasm that still contains an intron. So it turns out that in many virus-infected cells, we don't remove all the intervening sequences so that we can make different proteins from the same stretch of DNA. It's a process called alternative splicing we'll talk about in a moment. That is a conundrum, how to get unspliced or partially spliced mRNAs out of the nucleus, because conventional wisdom says that splicing is required for nuclear export of mRNAs. All right? That's what we learned years ago, but it turns out that viruses have circumvented that requirement. After all, it's just a human rule, right? We make up the rules and then we see that viruses break them. And so we'll talk later about how viruses get around the need for splicing to get out of the nucleus. All right, now let's take a look at that red arrow that I showed you on the initial double-stranded DNA, where I said this is where mRNA synthesis begins. And it's shown again here on this slide. At the top is a very short region of double-stranded DNA with all sorts of colors on it now. And at the right side, you see a red arrow. That's our transcription initiation site. This could be a cellular gene or it could be a viral gene, doesn't really matter. So right around the red area on the DNA is what we call an initiator sequence. It's a sequence of bases which, if you mess with it, you mess up where initiation starts. So this sequence specifies to the polymerase accurate starting points. We don't need to go into how it does that at this point. The point is just to show you all the different sequences that are located around the start site. If we go upstream, we have what's called a TATA sequence because it's TA rich. And this sequence binds a initiation protein called TF2D, and this is really important for transcription as well. These two sequences, the TATA sequence and the initiator sequence, constitute what we call the core promoter. You can take just that piece of DNA and put it in front of a gene, and you will get transcription under the right conditions. But there are other sequences as well that regulate transcription, okay? And they're also shown here. For example, upstream, there are local regulatory sequences, and we're just showing them as colored boxes. They bind a variety of other transcription proteins like the TATA binding protein TF2D. And you can see they can be anywhere from 500 to 100 to 500 base pairs upstream of the TATA sequence. So this core promoter is 20 to 35 base pairs long, and then a few hundred base pairs upstream are the local regulatory sequences. And those two parts, the local regulatory sequences plus the core promoter, constitute what we call a promoter, which gives you maximal regulation of the gene. Then there are some very distant regulatory sequences, which we call either enhancers or silencers, depending on whether they stimulate or repress transcription. These can be 100 to 10,000 base pairs upstream of the transcription start site. And they can influence transcription at a distance, and we'll show you in a moment how they work. So we have a very defined promoter region with regulatory sequences, but it can also be influenced by upstream sequences as well. And again, the key here is that all of these sequences, they bind proteins that regulate transcription. Transcription is a process that we regulate quite often, both in uninfected and virus-infected cells. It's a good way to determine what proteins are made at what time. It's very easy to regulate transcription because we have all these elements at which regulation can take place. Now, the cell has many situations where it needs to regulate transcription during development and growth and so forth, response to pathogens, but viruses also need to regulate as well. Viruses don't encode these core proteins that are involved in regulation, but they have their own proteins that can participate in transcription, and therefore they can regulate it as well. We'll talk about that today. Okay, so this is the basic concept I want you to get, that there's an initiation site for transcription, but there are surrounding sequences that bind proteins, and that can regulate the transcription. Because as you will see today, another key point is that during a virus infection, the making of proteins has to be regulated, and the easiest way to do that is to regulate transcription. There is some regulation that occurs at translation, but the vast majority is at the transcriptional level, and it's done by all of these transcriptional control regions. We don't modify the polymerase itself, we modify the proteins around the initiation site that are needed for transcription. So just as an example, I show you 3 different promoter regions from 3 different viral promoters. And they're both shown as our double-stranded DNAs. At the top is an adenovirus promoter called the major late promoter. This is a promoter that is very active late in infection. And you can see on the right is the transcription initiation site shown in the red arrow. You can see the initiator sequence around it. And upstream, there are some local sequences for the binding of a variety of transcriptional control proteins. Here they're called USF, USF1, and CPF. The names aren't important for you. Again, the point is that These are cellular proteins that are binding on the viral DNA to help stimulate transcription at that promoter. In the middle is the SV40 early promoter. This is a smaller double-stranded DNA virus. Again, you can see the transcription initiation sites at the right here. There are 3 different ones in very close proximity. This often happens. And you see, again, upstream regulatory sequences, binding sites for cellular proteins. And here, in this case, we have a repeated motif for a factor called SPL. And finally, at the bottom is an early promoter of adenovirus, the Adenovirus E2 early promoter. You can see it's different from the major late promoter. It has more initiation sites and binding sites for different cellular proteins. These have all evolved to give different levels of transcription. And as you will see, viral proteins are made that regulate transcription at these various promoters. So these are very much like cellular promoters. If you just look at the sequence and these binding sites, this could be a cellular promoter. But again, viruses do something different in order to regulate it. So here is a picture of how we think RNA polymerase II initiates transcription on a DNA template. And the DNA template is shown as a double-stranded molecule in light and dark blue. And this is the formation of an initiation complex. You can just see the yellow is the initiator sequence, and the red arrow, of course, is where mRNA synthesis will begin. So this complex includes a lot of different proteins. And you can barely see the RNA polymerase here in gray. It's in the back. That is the catalytic part of this complex. It is the enzyme that's actually going to synthesize the mRNA. But it needs all of these other proteins associated with it in this very big complex in order to make RNA effectively. And you can see they're all given various names. TF for transcription factor. When they were first purified years ago, they were just given numbers and letters, and nobody ever changed that. Here is TBP, the Tata box binding protein. Again, that is sitting on that TA sequence near the promoter. So all of these proteins, they interact with each other and with the RNA polymerase, the Pol2, and some of them interact with the DNA as well, but not all of them. And this is needed to get transcription. Again, this is a core set of proteins which every Pol2 promoter, whether it be eukaryotic, cell, or viral, needs to get mRNA synthesis. This slide, in addition, shows you how an enhancer works. So remember, enhancers are very distant sequences, up to 10,000 base pairs away, that enhance transcription. They enhance the amount of mRNAs produced. The way they work is that there are enhancer binding proteins in the cell which both bind the enhancers and bind parts of this initiation complex. It's a protein-protein DNA interaction. If you take out this enhancer, you get less transcription. You can still get it, but it's made more efficient when you have these enhancers present. And many viral genomes have enhancers in them as well. So an enhancer enhances initiation, not elongation. It enhances the formation of this complex and the initiation step. Now, all of these sequences around the promoter, including the enhancer, which you saw there, they're bound by sequence-specific transcriptional activating proteins. And these are the proteins that bind the DNA and in some cases interact with the RNA polymerase complex. And these tend to be modular proteins with very defined functional domains, as shown in this schematic of a protein. We have the N-terminus and the C-terminus. You can see there is a DNA binding domain. This is the part of the protein that clamps onto the DNA at whatever the specific is, whatever the sequence is that is specific for this particular protein. Then many of them have a sequence that enables dimer formation. These proteins often act as dimers, and if you can imagine that you have two proteins coming together, they clamp around the DNA, and the dimer sequence mediates that. And finally, they have what we call an activation domain, and that probably interacts with another protein in the complex to allow enhancement of transcription. So again, all of those various sequences I showed you next to the promoter, the USF sites, the CPL, the SP sites, they're all binding various proteins which have this modular formation. There are other kinds of proteins that also regulate transcription, and we call them co-activating molecules. So I've just told you about sequence-specific DNA binding proteins in the previous slide. The modular ones, where one of the modules is a DNA binding domain. And those can be host or virus specific. So some viruses do encode some of those that modulate transcription. But they're also what we call co-activating molecules that are virus specific. They don't bind DNA, yet they can still modulate transcription. And we'll look at an example of that in a moment. Many of these co-activators alter the structure of nucleosomal templates. Now, as you know, our DNA is very compact in the nucleus, and part of the structure is mediated by the wrapping of DNA over histones, which is shown schematically here. Each of these pink structures is a core histone around which the DNA is wrapped. And you may know that if the DNA is wrapped very tightly around the core histone, it's less likely to be transcribed. So if you think of this as a string wrapped around a spool, if you pull the string ends tightly, the RNA polymerase cannot, and all those accessory proteins cannot access the DNA template, so there's no transcription. On the other hand, if you loosen the thread around the spool of the histones, you get transcription. And we know modifications of the histones can either tighten or loosen the thread around the spool, and those modifications include acetylation of histones and methylation of histones. These are epigenetic modifications that regulate transcription. And we know that some of these viral co-activators, in fact, alter transcription by messing with the way that the histones are modified. Now here's an example of a viral protein that can influence transcription. And this is a really important one that we're going to come back later when we talk about how viruses cause cancer. So here is a schematic of the adenovirus DNA on the upper right. This is just a small portion of it showing one of the promoters. You can see the arrow, the red arrow for the initiation of mRNA synthesis. And this particular promoter has upstream a binding site for a cellular transcription protein called E2F. So these are cellular transcription proteins for which there are binding sites upstream of many promoters, and they're needed for activity. It so happens that the adenovirus genome has a number of these E2F sites, so it needs the E2F protein in order to get mRNA synthesis. Now, E2F proteins are these purple guys shown here in a cell, a resting cell. Most of our cells are not dividing, it turns out. Most of them are resting in the resting phases of mitosis. These E2F proteins are bound up by another protein called DP1 and a second called RB. So this complex, including the E2F protein, can bind the E2F binding site on the adenovirus genome. So you can see the E2F, the DP1, and the RB protein, the mustard-colored one. They're all bound at the promoter, but there's no transcription. And that's because the RB protein recruits histone deacetylases to the promoter. They remove acetyl groups from histones. This makes the DNA wrap more tightly around the histone protein, and it shuts off transcription. So the RB, the retinoblastoma protein, the way it's inhibiting transcription is by recruiting these histone deacetylases to the promoter and tightening up the DNA so the transcription doesn't occur. Now, of course, adenovirus, this is not a good situation for adenovirus, right? It requires these mRNAs to be made. So as you'll see in a moment, one of the first things that gets done in a cell infected with adenovirus is the synthesis of proteins, including one called E1A, early region 1A. I'll show you later where this comes from. That E1A protein, one of its functions is to bind RB. It binds RB, it prevents RB from sitting down on E2F, so RB can no longer recruit histone deacetylases to the promoter. And the effect is that mRNA synthesis can occur. So that's shown here by the color of the arrows. At the top, the inhibition of transcription by RB through the recruitment of HDACs is a red line with a flat end to it. That's typical for inhibition. And then on the bottom, we've removed RB, and now there's no more histone deacetylases. You can get transcription. So E1A protein, incredibly important protein to get rid of RB from this complex. If you take E1A away, the virus will not replicate. All right, and so this is a crucial modulator. It's an example of how you can modulate transcription. So normally, if E2F is not available, E1A can come in and make E2F available. That step, by the way, that liberation of E2F will come back to haunt us later on. Again, as I said, when we talk about how tumors are induced by viruses. So here we have a quiz. What is the first biosynthetic event that occurs in cells infected with DNA viruses? Membrane fusion, transcription, DNA replication, protein synthesis, or all of the above? A biosynthetic event. All right, what do we have here? 92% transcription. You have to make mRNA. You don't replicate DNA. That happens later. You need the mRNA to encode proteins that'll be used to replicate DNA. And we'll reinforce this at the end of this lecture. So it's transcription. And that repair that happens with those gapped genomes or the single-stranded genomes, we don't consider that a biosynthetic event. It's repair in order to get mRNA synthesis. All right, now where does the RNA polymerase come from? And what about accessory proteins like we've been talking about? So here's a table which lists the origin of the transcriptional components and particular viruses. So simple retroviruses, the transcriptional system, that means RNA polymerase II, and all the accessory proteins that I've been talking about, the DNA binding proteins, the co-activators, everything comes from the host for the simple retroviruses. You'll see later what we mean by that. There are retroviruses that encode just a few proteins and some that encode a lot of proteins. So the ones that encode a few proteins we call simple. They're not really simple. No virus is simple, okay? And I actually should never use the term simple because it's an insult to the virology, but simple simply means a small genome. They use all of the transcriptional components from the host. Then complex retroviruses, parvoviruses with single-stranded DNAs, papillomas and polyomas with double-stranded DNA genomes, but small genomes. These use the system of the host, but they make one protein, one viral protein that will regulate transcription. So it's amazing they can do that, and we're going to look at that in a moment. One viral protein will regulate transcription to time it as to the viral needs. The bigger DNA viruses, adenoviruses and herpesviruses, they, again, use the host RNA polymerase. So far, all these viruses are using host RNA polymerase, but they make more than one viral protein that can regulate transcription. And finally, we have the pox viruses, which encode their own transcription system. It turns out there are pox viruses and a couple of the really big viruses with huge genomes. They encode everything that they need. Why do you think, what would make a virus need to encode all of the transcription system? You know, this is a lot of stuff to encode, not just polymerase, but all the accessory proteins as well, all those proteins that bind around the polymerase, plus all the transcription factors. Why would a virus do that? Now, why questions are tough to answer. Let's phrase it another way. What would be the purpose of encoding your own transcriptional program? Think about where transcription occurs in a eukaryotic cell. Yeah?
Maybe it's too big to enter.
Well, most viruses are too big to get in, but the nucleus is where most eukaryotic transcription occurs, right? These viruses replicate in the cytoplasm. They don't want anything to do with the nuclear bureaucracy. They're out in the cytoplasm where there's no Pol2, there's no transcription proteins, so they have to make everything themselves. That's why poxviruses and these giant viruses encode the whole thing, because they stay away from the nucleus. Now, another key principle when we talk about mRNA synthesis is the idea that it's regulated. And I want to show you a couple of examples of how it's regulated. And you may ask right at the onset, why does it have to be regulated? Well, in a viral replication cycle, remember, just think generally, the genome gets in the cell. There would be no point in making structural proteins at that point, because you don't have any genomes to package. So you first have to replicate the genome, and then later you can make structural proteins. So there is always a series of events. One might say they're unfortunate, but there's a series of events that have to happen in order to make viruses. There's no point in wasting material at the beginning in order to, but you have to wait until things are available. So we regulate transcription in order to do that, and you're going to see how that happens in a moment. And here are two types of regulation that we see in virus-infected cells that are necessary for this. And at the top are autoregulatory loops. These can be either positive or negative autoregulatory loops. And here we have a positive autoregulatory loop in part A. So here we have a part of a viral DNA, and there's a promoter. The promoter site, of course, is shown in red. And that promoter encodes an mRNA for gene X. And the protein product of that mRNA happens to be a protein that stimulates transcription at the promoter from which its mRNA is derived. So it's stimulating its own synthesis. It's a positive autoregulatory loop. So as soon as that protein is made, you can make a lot more of the mRNA and a lot more of that protein. We see this often in viral replication cycles. As soon as the genome gets in, a protein is made which then positively regulates its own synthesis, so that can then go on and turn on a lot of other promoters as well. So that gets the whole system started very quickly. You can also have negative autoregulatory loops like this. So after a while, you've made a lot of protein X. Maybe you don't need it anymore. So protein X can then, under certain conditions, turn off its own synthesis. So you can have positive or negative loops. So that's one kind of regulation that we're going to see a lot of. On the bottom, in part B, is what we call cascade regulation. And this plays into the idea that you don't want to make all your proteins right away. Of course, if you only encode one protein, there's not much regulation you can do. But most viruses encode more than one. And as I said, you don't need to make structural components at the outset. You don't need those until you've made viral genomes. So the general scheme is to get in, make whatever proteins you need to replicate the genome, do some genome replication, and then make structural proteins and assemble them into particles. So here we have an example of how you do that. You have a viral genome with two promoters, a yellow one and a red one. And the red arrows, again, are the transcription start sites. The viral genome comes in the cell. You make mRNA encoding protein X. Protein X, in turn, is needed to get transcription at the promoter for gene Y. And so initially, maybe you have very low levels of protein X, so you don't have a lot of gene Y made. But then as you make more and more of X, you will get more and more of gene Y. And this can go on. And Y could be required for another set of transcription reactions as well. So that's what I mean by cascade regulation. You make one protein, and that protein happens to be a transcriptional regulator, and that regulates another promoter, and so on and so forth. And we're going to look at some examples of these now so that it'll be crystallized for you. So we're going to start with the simple viral genomes and get more and more complicated. We're going to look at three examples. SV40, shown here. We're going to look at adenovirus and herpes with increasing sizes of viral DNA genomes. All right, so this is the SV40 genome. We've seen it before. It's a circular double-stranded DNA. It's rather small, can't encode a lot of proteins, but it does pretty well. Now, the DNA has at one part a sequence called the ORI, the origin of replication. And this is where DNA replication initiates. We're going to look at this next time. But it's a multifunctional sequence. it also happens to contain two promoters. A promoter in one direction for the synthesis of early mRNAs, the one you need right away when the virus gets into a cell. And it also has a promoter in the opposite direction to make late mRNAs, which largely encode structural proteins. So the late one isn't on early. The early one goes on first. So at the top here is a timeline of what's happening after infection. So here at the left, we've infected the cells. The first thing that happens, this is a double-stranded DNA genome. You don't have to do any repair. First thing that happens is you get synthesis of early messenger RNAs, no late mRNAs. That synthesis is repressed by a mechanism that we'll look at in a moment. Only early, and one of the main proteins made early is shown here in blue, LT, large T antigen, or sometimes people just write it as a capital T. T stands for tumor. This was discovered because it was associated with tumors. And again, when we talk about how viruses cause cancer, we're going to really talk a lot about this protein. But large T is made very early on. It's made from one of these early mRNAs transcribed from the origin of replication. And it has a couple of important functions. First, as you will see next time, it's an origin binding protein. So it's needed to stimulate viral DNA replication. And this purple bar here on the timeline not only divides early from late phases of the cycle, but indicates the onset of DNA replication. So large T is required for DNA replication. You will see in a moment that DNA replication results in the turning on of the late mRNAs by a very interesting mechanism. Large T also participates in that mechanism, but eventually we don't need any more large T, so it can also repress its own synthesis, because late in the cycle we need mostly structural proteins. So the virus comes in, you make first large T, it turns on DNA synthesis, and then you get the late genes made. So this is a relatively simple cycle. There's just two phases, early and late. And again, early you don't need structural proteins. Early you're making what you need to replicate the genome, and in this case, that's just the large T antigen. Now, how does that late promoter come on? What makes it go on? It's very, very interesting. So here is, on the top line, the early phase of infection. This is the late promoter shown in red. It's the early phase of infection, but that red arrow denotes the late promoter. When this viral genome comes in the cell, remember the early promoter can work and get transcribed to the left, but the late promoter is silenced because there are proteins in cells called initiator binding proteins. They're shown in yellow. They bind to the viral DNA at and near the promoter, and they repress transcription. So these are repressive proteins. They are used by the cell to repress transcription under certain conditions. But the viral genome has binding sites for them. So when it comes into the cell, all the late promoters are silenced because these proteins are binding to them. Now, at this time, remember, early mRNAs are still being made. Large T antigen is being made. And what does T antigen do? It stimulates viral DNA replication. So as the DNA genome replicates, you get more and more of them, and that titrates out the initiator binding protein. The levels of initiator binding protein in the cell do not change. They remain constant. You always have a constant number of molecules. So as you make more and more viral genomes stimulated by large T, you titrate out the repressor protein, and now you free up the promoter so that you can make late mRNAs. It's a very simple mechanism. Clearly, the virus has evolved to take advantage of this for its temporal regulation. It has incorporated these IBPs around its late promoter so that they're silenced until they are needed. And as I said, these late mRNAs encode structural proteins of the virus particle. So that's really a very simple way of regulating gene expression. The early mRNA makes large T, the large T turns on DNA replication, and then DNA replication basically turns on the late promoter. So let's look at this in the context of an infected cell. Yes?
Is the IBP a cellular protein or is it a viral protein?
IBP is a cellular protein. And as I said, its levels are constant. It doesn't go up or down. So once it's all bound to the viral DNA, as you make more DNA, it gets titrated out. So here's a cell, an infected cell. We have a one-step replication cycle of SV40, this small double-stranded DNA virus we've been talking about. And these bind, of course, receptors. They're taken into the cell by transport on microtubules. They actually get out of endosomes and go into the ER, and eventually the DNA gets into the nucleus through the nuclear pore. So here in the nucleus is the viral DNA. And SV40 is very unusual. It's one of the few viral DNAs which is nucleosomed or chromatinized inside of the virus particle. So remember, DNA in us is wrapped around nucleosomes like thread around a spool. Most viral DNAs are not like that, but SV40 is. That's why we're showing. This is the incoming DNA at the top here, step 5. It's nucleosomed. All right, so that is now transcribed by host Pol II at the early promoter. You make the early mRNA shown here at step 6. That goes out of the nuclear pore into the cytoplasm. It's translated, and the main translation product is large T antigen. Large T antigen needs to get in the nucleus because it's going to stimulate DNA replication. So there we go, LT going into the nucleus, and arrow 10, it's stimulating DNA replication. Now we have red DNAs to denote that they're replicated, and they're also nucleosomed. And that's the early phase of replication. As soon as we have enough templates, DNA templates, to overcome the repression by IBP, we then go into the late phase of infection. We can make late mRNAs. And you can see step 11, we're making late mRNAs. They're being exported. And in the cytoplasm, they're translated to the VPs, the virion proteins, which are structural proteins. We can build new genomes. We can build new particles because we've replicated the viral genome. And eventually, the proteins in the DNA come together to make new virus particles. So it's just beautiful, as far as we can tell. You know, this is our human view of things, that it makes sense to make the protein first that you need for DNA replication, and then the structural proteins later, and it's probably right. And that's how it's accomplished. Now let's take it one step a little more complicated, just to show you that not everything is an early and late phase. And here we have adenovirus, transcriptional regulation. Now here we have 3 viral proteins that are going to regulate DNA synthesis and the transitions between the different phases of transcription. So for SV40, we had 1 protein that could do it. Here we have 3. The first one is E1A, which we've already talked about. E1A is an immediate early gene product. So here we have 3 phases: immediate early, early, and late. They could have called it early, middle, and late, but I think they discovered early, and then they realized there was one in front of it, so they called it immediate early instead of revising the whole scheme. As soon as the ad genome gets in the cell, E1A protein is made. And remember, this binds up RB and allows transcription at the viral promoters that require E2F proteins. So E1A is necessary for the transcription of all the early units. These early transcription units, and I'll show you what they look like in a moment, they all require E2F, and that's the function of E1A, is to free up E2F. So the early units now make another set of proteins. This is called the E2 proteins, and these are sort of like the T antigen of SV40. They are required for DNA synthesis and entry into the late phase. The DNA synthesis also titrates out repressors, very much like happens in SV40, which I told you that's what this means here. Anti-repression relief of promoter occlusion. To make more DNA, the repressors are titrated out. You then get late transcription activity. And finally, we do have another protein here, 4A2, another protein made from the early region that enhances late transcription. So it's a slightly more complicated series of events than SV40, and that's because there are more gene products involved, and you have to have a little finer level of regulation. Now here's the viral DNA genome. I just want you to look at this. You don't need to memorize any of this, except that there are early and there are immediate early, early, and late. So remember, the DNA shown here is a double-stranded DNA of about 40 kilobases. It's linear, and at the left end are the E1, the immediate early region, coding regions, and in this is the E1A protein, remember, which is needed immediately to free up the E2F factors. So The E1 is the immediate early, those are made first. And then we have early region transcription, and that's denoted by just E. And these green lines are the mRNAs. There's E3 and E4 and E2E, for example. And those encode regulatory proteins, as I've discussed. And then finally, we have the late transcription unit run from late promoters. And one of the important ones is the major late promoter. And it's called that simply because a lot of mRNAs are made at that promoter. And you can see these are structural components, the hexon and the penton that make up the particle. Now, what I just neglected to show you is in this early region transcription unit, the E2 unit. So first we have immediate early, then we have early. Here is encoded the DNA polymerase, right? So you need that in order to get genomes replicated, and you don't make late proteins until you have enough DNA replication, and then you can encapsulate those genomes. So initially, a little more complicated regulation early on, but then the middle phase or the early phase DNA polymerase, and the late phase the structural proteins. Now you can see here, the transcripts are complicated. They're scattered all over the place, and as you'll see in a moment, there's a lot of splicing that gives rise to these various different proteins. And here's an overall view of the adenovirus scheme, just to show the immediate early, the early, and the late phases. So the viruses get into the cell. We've talked about this entry step a lot for adenovirus. The DNA ends up in the nucleus. First thing that happens is you have an mRNA made, which encodes the E1A proteins. Remember, those E1A proteins have to come in the nucleus and bind to RB to free up the E2F transcription proteins that are then needed for the early phase of synthesis. And the early phase includes the synthesis of components needed for DNA replication, like the polymerase. All right, so the only promoter that works well on the DNA as it comes in the cell is the immediate early promoter. All the other promoters are silent until those immediate early promoter products, E1A, come on and allow the synthesis of the early genes. And then finally, after DNA has replicated, that turns on the expression of the late genes. So here's the late phase of transcription here. And these mRNAs are encoding structural proteins. And those again have to come back in the nucleus because that's where the viral DNA and the proteins assemble. So when you look at it this way, I think it makes a lot of sense to regulate transcription in this manner. The last one we'll look at is Herpesvirus, which is just slightly more complicated than Adenovirus and adds a twist that I want to show you. Here's the temporal sequence of events. You have, again, immediate early, early, and late phases, just like Adenovirus. The difference here is that the immediate early promoter, unlike that of adenovirus, doesn't work very well in a cell. So when this viral genome comes in, the immediate early promoter, which needs to be working in order to get these activator proteins like ICP0 to turn on early and late transcription, doesn't work well. And the solution is the virus brings in with it a protein called VP16. And this is different from any protein you see in polyomas, SV40 or adenovirus. This is an activator of this immediate early promoter. So that promoter on its own doesn't work well, but there is a protein in the capsid. It's actually just below the membrane of the virus and between the membrane and the capsid shown here on the upper left. It's called VP16. It activates immediate early transcription. These proteins, including ICP0, then control the synthesis of early and late genes as well. And again, we have DNA replication represented by the purple bar there. And when that occurs, that allows synthesis of the late mRNAs. And the replication proteins, very much like that of adenoviruses, are encoded in the early region as well. So it's a little more complicated, but the key difference that I want you to remember is the inclusion of this transactivating protein in the virus particle. And again, it's diagrammed here on this replication scheme. The viruses actually fuse at the plasma membrane, and all these little circles are the VP16 protein coming into the cytoplasm out of the particle. They get in through the nuclear pore. They're imported specifically, VP16 into the nucleus, and of course the viral DNA gets in the nucleus as well. The VP16 allows efficient transcription of immediate early mRNAs, and those encode proteins that are then needed for early gene mRNA transcription, which would include the proteins you need for DNA replication. And then finally, the DNA replication relieves occlusion of late promoters. You get late proteins which encode structural proteins. So it's a very interesting combination of transcriptional regulation and DNA synthesis to get the proteins when you need them. Next one is Adenovirus E1A protein stimulating the expression of adeno E2 protein, which then stimulates adenovirus 4A2 and L4 protein, is an example of a negative autoregulatory loop, repression of gene expression, cascade regulation, or dimerization. All right, that's an example of cascade regulation, one stimulating the other, stimulating the other. There's no repression at all involved here. All right, let's look at some of the processing events that happen on the mRNA. The first thing is the addition of this cap structure. So here on the right is base 1 of the mRNA, base 2, and you see they're connected in a 5' to 3' linkage. The 5' carbon on the ribose is attached to the 3' hydroxyl. However, the cap is added in a backwards direction. It's 5' to 5'. That's one reason why we call it a cap. Not only is it attached in a 5' to 5' linkage, there are 3 phosphates in between the 2 bases instead of 1 in the rest of the RNA. And some of the bases are methylated. You can see up here on the upper left, there's a methyl group. On base 1 of the RNA and base 2, there are also methyl groups. So that's a cap structure. It's needed for efficient translation. As we'll see, it's needed for stability of the message and a variety of other features as well. So this addition of the cap is part of the transcription process. When we have a double-stranded DNA with a promoter, remember the RNA polymerase II assembles at the promoter at the initiator sequence and begins to make a transcript. After about 20 or 30 nucleotides, then the cap is added. It doesn't happen initially, only when you have a nascent transcript shown in green. There are signals given to the enzyme by phosphorylation, and that results in capping of the transcript. So capping is a co-transcriptional event. At the other end of the message, you have to add a poly-A sequence. And the way that happens is that messenger RNAs have at their 3' ends a poly-A addition site, which is a very specific sequence, AAU, AAA, or AAT, AAA. And that poly-A addition site is recognized by a variety of cell proteins, which are all shown here as various colored spheres and ovals. One of them is an enzyme, poly-A polymerase, and the other one is a nuclease that cleaves just downstream of this poly-A addition site. So if you look at 5 lines down here, you see now this is a nascent transcript now. We have a mRNA being transcribed by Pol2. As soon as the poly-A addition site is recognized, that transcript is cleaved. But you can see that the Pol2 has gone beyond the poly-A addition site. So we've got extra sequence at the 3' end. The cleavage occurs right 3' of the poly-A addition site, and the remainder of that RNA is presumably degraded because it's not needed. And then the enzyme poly-A polymerase adds 200 or so A's to the 3' end. So polyadenylation is a post-transcriptional event. The polymerase has gone by the addition signal for poly-A, and then only later does cleavage and polyadenylation occur. Now, let's compare the different ways of putting poly-A on viral mRNAs, because last time when we talked about RNA viruses, we talked about how they add poly-A, and they're all summarized here. So what I just told you is called post-transcriptional addition of poly-A, because the transcript goes beyond the poly-A addition site, and then it's cut, and then poly-A is added. Cleavage of pre-mRNA followed by polyadenylation. So the enzymes that do this are cellular, and this happens for all the viruses that we talk about. Hepatitis B, herpesviruses, polyomaviruses, retroviruses, as we'll see as we go along. In contrast, the RNA viruses that we talked about last time, they typically add poly-A during mRNA synthesis. For example, reiterative copying of a U stretch in the template for influenza virus or VSV. Remember, the enzyme stalls at a U stretch, and we talked about two different mechanisms for VSV or flu for doing that, and then it stutters and it makes a poly-A. Well, in some viruses like polio and flavies and togas, there's a U encoded in the RNA, and the polymerase simply copies that. So very distinct mechanisms for making poly-As. Okay, which statement about polyadenylation of DNA virus mRNAs is correct? It always occurs in the cytoplasm. It occurs after cleavage of pre-mRNA. Poly-A is added to the 5' end of pre-mRNA or is specified by a stretch of U residues in the template. All right, a little uncertainty here. 60% of you said it occurs after cleavage of pre-mRNA, and that's absolutely true. Doesn't occur in the cytoplasm. This is a nuclear event, just like splicing, polyadenylation. All this processing occurs in the nucleus before export. Poly-A is added to the 5' end. No, it's added to the 3' end, specified by a stretch of U residues. That's only for RNA viruses, right? And this question is saying DNA viruses. Let's finish by talking a little bit about splicing and its regulation. Splicing of mRNA precursors was discovered in cells infected with adenovirus. Why? Because you can make pure DNA and you could make pure mRNA at a time before cloning. Now today we can do it with any gene, but at this point you could only do this with viruses. Here's the viral DNA shown by the two strands of DNA. Now these investigators who did this, including Phil Sharp and Rich Roberts, and they got the Nobel Prize for this years later, they wanted to know where the adenovirus mRNAs came from on the viral genome. So they were using electron microscopy. They would take the viral DNA and separate the strands and take purified viral mRNAs and hybridize them and look at them under the microscope. When you hybridize the two sequences, you get a double strand, which you could recognize in the electron micrograph. And if they weren't hybridized, they were single-stranded. So on the right is a tracing of the actual electron micrograph. It's a little complicated, so I prefer to draw my own, which is shown here. So here's the result they found. They took the late major mRNA of the virus, which encodes the structural proteins, and that's shown in green. And they hybridized that to the viral DNA. And they kept seeing these loops, like 3 loops in this one. 1, 2, 3. The implication being there's more sequence in the DNA than in the RNA. What did that mean? They thought about it, and they finally said something must be getting removed from the mRNA, and this is where splicing was discovered. So basically, the pre-mRNA is a direct copy of the DNA, but pieces get spliced out in the nucleus so that the final mRNA is shorter, and that's why you get these loops in the electron micrograph. And the late mRNAs have this structure. They have intervening sequences that are removed by splicing. Since then, we know that splicing occurs in many, many cellular mRNAs as well as viral mRNAs. And people have studied this a lot to understand how it happens. And a lot of that data are shown here. Here at the top is a depiction of the sequences at the junction between the areas that are going to be spliced out, which are shown in pink, and the rest of the mRNA. So we have come to call the spliced out parts introns, or intervening sequences, and then the parts that remain in the message we call exons. So at the top we see exon sequence, then we have a 5' splice site and a 3' splice site, and of course the 5' will be joined to the 3' and the pink part will be thrown away. And what they've done on this picture is show the sequence consensus at every base at the 5' splice site within the intron and at the 3'. And you can see there are some bases that are very conserved if you look at many, many different mRNAs. For example, this A residue in the middle of the intron is 100% conserved in every mRNA precursor that you look at. And this led to the solution of the actual mechanism of splicing, which is really cool. So here we have on the top the linear version. But what happens in the splicing, there's a first step where that A, the hydroxyl of that A, attacks the phosphodiester bond at the 5' splice site. That releases the pink intron as a lariat. So now you see it looks like one of those ropes that the cowboys throw, a lariat. You have the free 5' end of the 5' splice site. It now has a 3' hydroxyl that attacks the 3' splice site. It breaks that phosphodiester bond, and now you have the 2 exons spliced together. And what's released is this excised lariat intron. So you have a chemical reaction, and you may be thinking, how does this happen so that the 5' and the 3' are in the right place? Well, it happens on a structure called the spliceosome, which is shown here. The spliceosome is a ribozyme. It's composed of RNA and proteins, but it's the RNA that carries out the catalysis. So here's our pre-mRNA at the upper left. It is bound by a number of what we call small nuclear ribonuclear proteins, or SNRPs. These are RNA protein particles, and you can see them outlined here in circles. The circle is the protein, and the colored region is the RNA. These RNAs base pair with the conserved sequences at the junctions and within the intron, and they direct all these reactions that I just showed you. For example, you can see all of these base pairing interactions between the intron and the RNAs of the spliceosome. And that transesterification reaction, that first one where the A in the intron, which is up here, I can't really see it, I think it's right there, that attacks the 5' splice site that's going on here. It's all held in place because these RNA protein complexes are holding it in just the right way based on complementarity between the RNAs. And eventually that bond is broken. You can see the 5' splice site is free in step 4. It's attacking the 3' splice site. They're ligated together. Now we have the mature mRNA and the lariat intron, which is discarded. The key here is that we have splicing occurring on a very large protein RNA complex called the spliceosome, which makes sure that all the places are in the right parts in order for this to happen. Now, because of splicing, you can get different genes from different proteins made from one gene by a process called alternative splicing. For example, if you have 2 introns, as shown at the left here, you could splice both of them out and make a single mRNA that encodes a protein. But you can do all kinds of alternative splicing. You could remove one intron or the other. You can do alternative 5' splice sites or alternative 3' splice sites. And in every case, you get a different protein. So you can see right away why this has evolved in most organisms, because it's a great way to diversify your genetic complement. You can make a lot of different proteins by just splicing in different ways. Here's what adenovirus does, for example. You've seen the double-stranded DNA of adenovirus. Here is the major late transcription unit. It's a really, really big mRNA that goes for almost the entire genome, starting at the major late promoter. It's capped and polyadenylated. It's alternatively spliced. It's got lots of introns, which are shown in pink there. It's alternatively spliced and alternatively polyadenylated. There are lots of different poly-A addition sites, so that in the end, you get all kinds of different proteins from this one gene. You get, for example, a number of late proteins that are needed here. And look at all these structural proteins, the 4-hexon protease, etc., all from one gene. Okay, so the only way you can do this is by alternative splicing and alternative poly-A addition. Now, in cells, such as our cells, when mRNAs get spliced, they then become marked for export out of the nucleus. Remember, they're all spliced in the nucleus, and then they go out the nuclear pore into the cytoplasm. The assembly of the spliceosome on the pre-mRNA leaves proteins behind, which are then recognized by the nuclear export machinery. So that's what we're showing here. At the top is an intron-containing mRNA, which is bound by all sorts of proteins during the splicing process. We now have splicing occurring, and then some of these proteins are left behind from the splicing process. They are recognized by components of the nuclear export machinery. So all the splicing, of course, is happening in the nucleoplasm. And one of these export machineries proteins is shown here, NFX1. It recognizes this mRNA and it goes, aha, it's been spliced, now it can get out. Because in our cells, if you're not spliced, you don't leave the nucleus. It's another kind of quality control check. Right? So splicing marks mRNAs for nuclear export. The problem is there are lots of viruses that don't completely splice all of their mRNAs. So how do they get around it? Here's how they get around it. On the right, again, is that same scheme that I just showed you, that mRNAs are marked for exports by being bound with proteins that are recognized by the export machinery. Here on the left is a retroviral RNA. It is not spliced. There's an intron there shown in gray. Now, this will eventually get spliced, but a certain fraction has to remain unspliced so that a different protein can be made. How does this unspliced messenger RNA get out of the nucleus? Because according to the rules on the right, it shouldn't. Well, the 3' end of this mRNA contains a sequence called the constitutive transport element, or CTE. And guess what? CTE binds components of the nuclear export pathway. That NFX1 protein, which was bound to our cellular spliced mRNA, that recognized the CTE, and it's like a ticket for getting out of the nucleus. The export machinery says, this mRNA is okay, it must be spliced, but of course it isn't, and it gets out. So I think that's really cool how these have evolved so that they can export both spliced and unspliced messages out of the nucleus. There's one more example I want to show you. It's HIV, one of these retroviruses with complex genomes. They encode a lot more proteins. They make a very long transcript, which gets spliced, but they also need to make a variety of transcripts that don't get spliced at all. And in fact, one of the things they want to do is export this fully unspliced mRNA. Now, they of course have another mechanism for doing it. It's slightly different from the retroviruses I just showed you. What happens initially after that transcript is made, it's completely spliced all the way down to a very small mRNA which gets exported because it's marked by splicing for export. It encodes a protein called Rev. Rev goes back in the nucleus and it binds to a sequence at the 3' end of unspliced mRNAs, and it allows them to be transported out because Rev is recognized by the nuclear export machinery. So 2 ways to circumvent the need for being spliced for export so that you can move out partially spliced mRNAs. And of course, that gives you more flexibility. All the adenovirus mRNAs I showed you that are partially spliced and give you more coding capacity, that's how you do it. Splicing is value added. You get new genes made by alternative splicing. You can expand the coding region of a small genome, and you can regulate gene expression. You can regulate when proteins are made or not made by splicing that gives rise to them or not. So remember, to make mRNA, you need a double-stranded genome. If it's not double-stranded, if it's single-stranded or gapped, you have to first repair it, and then you can make mRNA. So why is it so important to make mRNA first? That's because viral DNA replication always requires the synthesis of at least one viral protein, and sometimes many more, and that's why you have to make mRNA. You can't replicate your genome until you make at least one of those proteins. And next time we're going to talk about how you take these viral proteins and use them to replicate DNA.